So, I want to tell you a story. It's a story of resurrection, really, about bringing a plant back from the absolute brink of death. And this isn't just a simple gardening tale. Oh no, this is a deep dive into chemistry. It's a high stakes rescue mission for one of the most beloved and, let's be honest, sometimes frustrating houseplants out there, the orchid. If you've ever had an orchid, you know this exact feeling, don't you? That sinking in your stomach. You lift the plant out of its pot, hoping to see those beautiful silvery green roots, but instead, you find this, a soggy, black, hollowed-out mess. The source material calls it the black collapse, and yeah, it really feels like game over. But here's the twist. What if that moment, that feeling of total failure, is actually the starting gun for an incredible comeback story? What if, with a little bit of science that you probably have in your house right now, we could turn what looks like certain death into a dramatic revival? That's exactly what we're going to break down. Okay, so to be the hero in this story, we first have to really understand the villain. And in this case, the villain has a simple, nasty name, root rot. So what is root rot, actually? It's way more than just the roots being too wet. The real problem is that they're oxygen-starved. See, when the potting mix gets all compacted and waterlogged, air can't get in. The roots literally can't breathe, so they start to decay and ferment. The plant is, and this is wild, drowning and suffocating at the exact same time. Let's just put these side by side for a second. A healthy root, it lives in an oxygen-rich, aerobic world. It's firm, it's a healthy color, and its job is to pump water up to the leaves. But a rotting root, it's trapped in an oxygen-starved, anaerobic nightmare. It turns to mush, goes black, and its function completely reverses. It's no longer feeding the plant. It's become food for pathogens that are eating the plant alive. All right, with the enemy clearly identified, it is time to reveal our secret weapon. And it's not some fancy, expensive chemical you have to order online. In fact, it's probably sitting in your medicine cabinet right now. Yep, that's it. Good old hydrogen peroxide. The stuff in the brown bottle. This molecule is basically just a water molecule with one extra oxygen atom kind of clinging on for dear life. And that unstable extra atom, well, that's the key to this whole operation. So you've seen it, right? You pour it on a cut and it foams up like crazy. But why? Well, when hydrogen peroxide touches all that organic decay, our rotting roots and the nasty bacteria feasting on them, it meets an enzyme called catalase. Catalase is a super powerful catalyst that just instantly rips that extra oxygen atom right off the peroxide molecule. And what's left? You get plain water and a massive, fizzing, bubbling release of pure oxygen gas. That's the foam. And this is the most crucial part. That foam isn't just for show. It's a legit two-pronged attack. First, it's a physical assault. The force of all those tiny oxygen bubbles is like a microscopic power washer, literally blasting away dead, mushy tissue and clumps of germs. Second, it's a chemical weapon. Those microbes causing all the rot, they evolved to live without oxygen. So a sudden massive blast of pure oxygen is completely toxic to them. It annihilates them. Okay, so knowing the science is one thing, but now we have to apply it. We're moving from the chemistry lab straight into the operating room. This is where it all becomes a very real, very precise surgical procedure for our little plant patient. And the protocol here is really specific. Step 1, total debridement. You get some sterile scissors and you have to be absolutely ruthless. You cut away every single bit of mushy, hollow, dead tissue. Then, step 2, the peroxide bath. You pour standard 3% hydrogen peroxide right on there and just watch the magic happen. It foams and sizzles, cleaning everything your blade couldn't reach. And finally, step 3, a gentle rinse to wash it all away. And let's be perfectly clear about what we're doing here. We are not just cleaning the plant. We are fundamentally resetting its entire environment. We're taking it from a state of oxygen-starved decay and creating a sterile, oxygen-rich foundation where new life actually has a fighting chance to begin again. Now, this is where it gets truly amazing. Because the rescue operation doesn't just stop the rot, it actively signals to the plant that it's time to start living again. Think about it. After being starved of oxygen for who knows how long, that sudden, massive flood of it from the peroxide reaction is like a jolt of lightning to the plant's whole system. It's a powerful, biological wake-up call. But why is it so effective? Well, because oxygen is the key ingredient for creating something called ATP. And ATP is basically the energy currency for every living cell on the planet. By flooding the plant with oxygen, we're essentially handing it a huge bag of cash, giving it the fuel it so desperately needs to start the incredibly hard work of regrowing itself. But wait, there's more. There's this fascinating internal chemistry going on too. When the main roots die off, the plant's whole hormonal balance is thrown into chaos. The hormones that were preventing new roots from growing are suddenly gone. So these other hormones, called cytokinins, they take over and send a signal to dormant little growth points on the stem, basically screaming, "Emergency! We need new roots! Grow now!" And unbelievably, they listen. So the surgery is over. The signal has been sent. The plant is ready to fight, but now the patient needs intensive care. The hard part isn't over yet. A rootless orchid is incredibly vulnerable. I mean, think about it. It has no way to drink water. So you have to create a plant ICU. And the sphagnum bag method does exactly that. By resting the orchid on some damp sphagnum moss inside a sealed bag, you create this super humid little world where the plant can absorb moisture through its leaves just to stay alive. And when I say humid, I mean humid. We're talking over 80% humidity. This is totally non-negotiable. Without it, the plant is just going to dry out and die long before any of those new roots even have a chance to start. This one number is the absolute key to post-op survival. And then, after weeks, maybe even months of patient waiting, you might just see it, the ultimate trophy of this whole battle. That first, tiny, vibrant green nub of a brand new root pushing its way out into the world. That right there, that is the moment you know. The rescue mission was a success. The patient is gonna make it. So what's the real takeaway from all this? It's just a powerful lesson in resilience, you know? A plant that looks like it's on the edge of death isn't necessarily gone for good. It might just be trapped in the wrong chemical state. And by understanding the science, we can step in and, as the source so beautifully puts it, give it the oxygen it needs to remember how to live.